I want to thank the organizer so much for giving me the chance to speak to you all. This is a really different audience than I normally speak to in terms of the breadth of expertise I think will be in the room. I've tried to pitch the talk to be understandable by a general audience, but I am a basic scientist, so there will be some graphs. But I hope that those will be clear enough for everyone to understand, but please do ask questions. I'm going to tell you two stories. They're both fairly short, and I think I've timed the talk to be about 40 minutes. So hopefully we'll have plenty of time to chat afterwards. I'll just start by saying that I am a basic scientist. I'm not a clinician. None of the information in this talk is meant to be clinical advice. But I do think it's important that we start thinking about the gap between the clinic and the basic science. And so I thought that would be the perspective that I bring. I titled the talk, When Scientists Learn from Patients, What Do Viral Pandemics Teach Us About Fungal Infections? And I'm going to talk to you about that in the context of two pandemics. One is the HIV pandemic, which we are now 41 years into and still fighting. And the other is the ongoing COVID pandemic, because I think these are both sort of global world events that have impacted us all in ways that we may or may not appreciate. So we're going to start with the Cryptococcus story. I've started this illustration to kind of bring the whole thing together, because when we think about microbiology, what we're really talking about is our interaction with the organisms that surround us all the time. And I'm showing you here the organisms that I care about. Cryptococcus neoformans, which you can see in the bottom here in this interacting with the tree. We normally see it associated with trees or with birds. There's also an association with hyrax, which is this little creature here. And it's really soil-associated organism. And the other story that we'll talk about is a story about mucormycosis, and that's represented by these organisms here. And my favorite representative of this group is called the Rhizopus species, and we'll talk about that in a bit. So let's just get started with the Cryptococcus side of things. So I just want to set the stage here. It's 1981, and you're an infectious disease specialist. You see infectious diseases coming into your clinic fairly often, tuberculosis, things like that, right? And you start seeing a lot of patients coming in with very unusual diseases. And the two ones that are really surprising to you are ones called PCP, which is pneumocystis pneumonia, and the other one's called Cryptococcus neoformans. And this is the first signal to infectious disease doctors that something has really gone wrong, because these are diseases that we don't normally see in patients. And now suddenly large numbers of people are coming into clinics with these infections. So this is an example of a patient who you might have seen in the clinic. This is a biopsy from an American man who died of AIDS in 1983. And what you can see in this tissue section is actually this organism, Cryptococcus neoformans. And I want to point out a few features of this. You can see these nice round yeast. Hopefully you can, I'm not sure if my pointer is coming through. Let's see, laser pointer. So you can see these nice round yeast. And they also have a lot of different colors to them, right? Some of them are very dark and some are these halos. And that's because they have differences in their cell walls between the individual cells. Oh dear, now I've made it difficult for myself. All right. And this is important because what it's telling us is that Cryptococcus is actually an early indicator of HIV infection. And this is something that continues to this day, where patients coming in with Cryptococcus neoformans infection make up 60 to 70% of patients with HIV. And this is really the thing that tells doctors that something is wrong. It continues to this day. This wasn't a new organism. We'd known about it for many years. This is what people normally looked at when they were thinking about Cryptococcus neoformans. And this is an example of that growing on an agar plate. It's a budding yeast. So this is very similar in appearance to the organism that makes bread, but it's very different species. You can see that this is an organism that's budding yeast here. You have your mother cell and your daughter cell. These are nice homogeneous population going through their cell cycle in a very regulated way. And there was a significant amount of work done to understand these guys. And this is the way we were really thinking about them for a number of years. The problem is that when we're looking at patient samples, that's not what we saw. Instead, we would see this really mixed population of small and large cells, cells with differences in their cell wall, like I just showed you, And these cells could get really, really big. So here I'm showing you a scale bar that's 40 microns, and that's a cell that's even larger than that, whereas the cells in the previous picture were about 5 microns. So they were more similar to like this size here. So we have a lot of differences in patient samples compared to what we are seeing in the lab. And for a long time, before the AIDS epidemic and even into the 2000s, this was sort of chalked up as this is a weird patient sample that doesn't represent what most clinical isolates are going to look like, and so we don't really have to worry about it. And one of the things that was a telling point that maybe that's not true was this work that was done back in 1973 by these scientists on this sample that came from a young Zimbabwean woman who came in with an abscess in her lung. So when the scientists in 1973 took those cells from that abscess and they put it on standard growth media in the lab, it went through its normal cell cycle and it looks just like the yeast that the movie that I just showed you. And they thought, well, that's weird because that doesn't look like what we saw in the patient. So they took those cells and they put them back into a mouse and they looked at they looked in the mouse and they looked again very, very large like they did in the patient. So there was some kind of hint here that maybe what was happening in the humans and in the mice wasn't really representative of what was happening in the lab, and there was a disconnect there. And this was kind of ignored for 30 years until the HIV epidemic hit, and we started seeing large numbers of patients with these intractable infections. So in 2001, a group in the US led by Arturo Casadevall repeated this experiment. They took yeast that were growing in their lab and they put them in mice, and they were able to show that once again, they got these huge, huge cells that they called giant morphology. And again, this was sort of said, okay, well, that's a weird observation, but we really don't know what it means, so we're just going to let it sit for a while. And it sort of percolated among scientists for a number of years until in 2010, 2 labs demonstrated that actually this is a real thing that these fungi do. And they characterize these guys. They called them Titan cells because one defining feature of them is that they're huge. They're just so, so big. So they thought the word Titan characterizes that. So you can see an example of that here. This is a tissue section from a mouse. And you can see, again, just like in that biopsy from that patient, we have these very large cells with different cell walls, and we have little tiny cells like we would see in the lab. The other thing that was really unusual about these was that they would produce daughter cells that are much smaller than the mother cell. So here you have a large mother cell and a small daughter cell. And that's different from the movie that I showed you where everyone's exactly the same size, right? So what this is telling us is that these in vivo cells, these Titan cells, are a true phenotype, and they have distinct differences from what we would see in the lab. They did a lot of other work to characterize what these were, and they made some definitions for them. So what they defined these cells as, they're larger than 10 microns in diameter, so that's twice as big as a normal yeast cell at least. They are highly polyploid, meaning they have multiple copies of their genome. They have this really thick cell wall, which you can see very nicely in this picture. And they have an altered capsule, and I'll show you a picture of the capsule in just a minute. And they also were able to show that these cells were really important for disease because they are associated with evasion of the immune system. So these are immune cells that we're going to see quite a lot in this talk. These are called macrophages, and they're what are called phagocytic cells. What they do is they go around eating things, right? And by eating them, they can digest them and they can kill them and they remove them. And that's a front line. It's a really important first step in our immune systems protecting us from infection. So you can see here an example of macrophages that have inside of them yeast cells, normal sized yeast cells, which they've eaten and they're killing. But you can see that that Titan cell is way, way too big for them to kill. So this is going to be a way for the fungus to evade the immune system. So these Titan cells are associated with macrophage evasion, a non-protective immune response, and they actually get to the brain faster. And that's associated with a poor outcome for patients. So not only is this a weird feature that we only first observed in patients, not just a weird feature, but it's actually contributing directly to disease. So that's a really, really important finding. The issue is that we were only able to study these in mice. And that's a really hard thing when you're a basic scientist because it's expensive. It's very challenging to draw specific conclusions about something that you can't see. And so it was a major challenge in the field, which is how do we go from this in vivo or this in mouse phenomenon to an in vitro or in lab model that we're actually able to investigate. And I'm going to tell you a little story about how we managed to do that. This all started with an undergraduate who was in my lab named Tom Drake. And Tom was really interested in studying capsule. And I'm sorry, I haven't shown you a picture of that, but you'll see a picture of that in a minute. And one of the ways that we study capsule is we grow cells in CO2 and concentration that mimics what this concentration of CO2 is in our lungs, because we know that when we breathe cells in, they make a capsule, and that oxygen concentration in our lungs, the CO2 concentration in our lungs, is one of the major signals for that. And Tom was interested in understanding capsule, but he was growing his cells in a little slightly different way than we would normally do it. Instead of using a rich medium, which is the standard approach of basic scientists, because our philosophy is we want our cells to be happy before we put them under stress, because if they're not happy before we put them under stress, then they're going to die, right? So make yourselves happy, give them plenty of sugar, and then put them under stress. And Tom took a different approach, which he grew his cells under minimal conditions and then put them under stress. And this was an experiment that we talked about before I went to a conference. I got a message from him when I was standing in the back of the room listening to somebody else talk, and I checked my mail. And this is the picture that he sent me. And what I hope you can see is, so there's the fluffy capsule that we wanted to see, he wanted to study, right? But then he said, you know, I think there's something wrong because these cells are really big. They're way too big for what I wasn't expecting. And what I hope you can tell is that Tom figured out how to grow Titan cells in vitro. And this was a major discovery for the lab and for the field, and really changed what we're able to do. So what you can see here is a yeast cell, small yeast cell, and that white halo that's around it. That's a sugar capsule that protects the cell from phagocytes, for example, and from other stress conditions. And in the larger cell here, you can see what's a very nice example of a Titan cell with its very, very thick cell wall. Its capsule is denser, so hopefully you can see that even though the cell is really big, the diameter of its capsule is the same as that little tiny cell, so it's a much more compressed capsule. And it also has this big vacuole in the middle. These are all very clearly defined features of what a Titan cell looks like. Okay, so that's great. We can grow Titan cells in the lab, but what can we actually learn from this model? Well, the first thing we can do is take some movies. And I hope you can see that our Titan cell here, going from a yeast into a Titan cell, is very different in the way that it grows from the movie that I showed you previously. So you have this asymmetric where the daughter cell is smaller than the mother. It's actually a lot faster too. So while yeast cells go through their cell cycle every 2 hours, this goes every hour, like clockwork. It's very nice and fast. And the other thing that you can see is that there's a lot of heterogeneity there. There's a lot of different cell sizes. And I think you can see that really nicely in this picture here, where you can see the large Titan cells. We've got our yeast cells, our standard yeast cells in the upper left-hand corner there next to the Y. And we have these little tiny cells. These little tiny cells hadn't been described before, and I'll tell you about them in a little bit. So what can we learn from this in vitro model? Well, the first thing we had to do was actually replicate the experiment that Tom did, which was not as easy as it might have sounded, because when you're just doing a happy accident, a lot of times you're not thinking very carefully about what the experiment that you're doing is. But the first thing we noticed was that one thing that Tom did really differently than normal was he grew his cells under minimal media, and he also used a really low amount of cells to start his culture. So I said before that when you're a basic scientist, you want your cells to be happy and you want to put enough cells in there so that you can do your experiments easily. So normally you would fill your flask with cells, and if you did that, you would see yeast. But Tom, being a novice, started off with just one cell, basically. And when we looked at that, he saw these really, really big guys and this heterogeneous population, right? So just by changing some really basic parameters of this experiment, suddenly we see a lot of variety. So what is going on here? Well, one thing that is clear is that Cryptococcus knows how many neighbors it has. So if you start a culture that's crowded, it makes a different kind of yeast than if you start a culture that's not crowded. And we can actually do an experiment where we use half, again, as many cells. And the prediction would be that you would see increasing cell size. And when we do that experiment, that's exactly what we see, right? So we go from a crowded culture to an uncrowded culture, and you can see that the size of the cells in that population is getting bigger. That's kind of crazy to think about, right? How does a fungal cell know at the beginning of an experiment what's going to happen at the end? There's some really cool questions you can ask there. One of the things that this tells us about is a process called quorum sensing, which is a really important process in bacteria, but isn't very well studied in fungi. And this is where it's going to get a little tiny bit technical, but I hope that it's going to be clear. So quorum sensing is a way that cells communicate with each other. Basically, cells can put molecules out into their environment, and then their neighbors can detect those molecules, and they can make decisions based on how much of that information they get. So here I'm showing you a little picture. So here's a fungal cell producing its quorum sensing molecule. Here's its neighbor binding that molecule and then creating, making a decision based on the ability to sense that molecule. So when there's a lot of cells around, there's lots of this molecule around, and the cells say, oh, don't do the process, don't make Titan cells, let's grow as yeast. But if there's only 1 or 2 cells around, then this molecule isn't detected, and instead the cells can make Titan cells. So again, this is telling us coming from a patient observation, it tells us about the basic biology of these organisms, their ability to sense their neighbors. What else can we learn from this model? Well, we know that these guys are highly heterogeneous, right? So one of the things that we were really surprised to see, I'll just go back and show you again, we have these little tiny guys, right, that hadn't been described before. We know Titan cells are in vivo, but what about these little tiny cells? And when we looked more closely at these, we were able to say, yes, these are definitely a distinct cell type that have a lot of features that make them really unique, even from yeast. So I'm showing you transmission electron microscopy here, which allows us to see a lot more detail than just light microscopy, which is what I was showing you before. And you can see here's a beautiful example of a yeast with its nice cell wall. It's got its nucleus. You can see a lot of little organelles in there. And when we zoom in, you can see here's this beautiful cell wall with layers of the molecule that makes up the cell wall. At exactly the same magnification in a Titan cell, you can see that cell wall is super, super thick, right? Much, much more dense than in the yeast cell. And here's the overall picture of a Titan cell. Again, it's mostly vacuole, right? There's not a lot of information in there. This third cell type, which we're calling a titanide, because they're just riffing on this Greek history here, is very different. It's oval in shape. It tends to be very thin cell wall. And whereas the Titan cell has this big vacuole, again, you can see here's the nucleus, lots of nice little vesicles and organelles inside of the cell. So we think it's really a distinct kind of way of being. And I'm just quantifying that cell wall difference here, where you can see the yeast have a particular thickness of cell wall. The Titan cells tend to have much thicker cell walls, and the titanide cells tend to have much thinner cell walls. All right, so this all tells us that rather than these Titan cells being a weird thing that was happening in this one patient that came out of Africa, it is instead a basic way of being for this organism based on the information that it's receiving from its environment. It's in a low density, there's oxygen limitation, there's nutrient starvation. So what are the signals that this fungi starts responding to form these cells in the environment? So we went back to thinking about, well, where exactly is Cryptococcus in the environment? And I just remind you, my little picture here, we've got our birds, we know it's associated with pigeons, we've got our hyrax, it's often found in arboreal environments. And we did a lot of work to try and figure out what might be the signal, but what we were forgetting was that these are soil organisms, and what else is in the soil? Obviously other microbes, right? So I'm showing you here bacteria, which if we weren't fungal people, we would have all stood up and said, obviously it's bacteria, right? Because bacteria are everywhere. It turns out that bacteria are one of the major signals that are causing Cryptococcus to form these Titan cells. And the proof for that is right here. So here we have our normal way of making Titan cells, which we use something called fetal calf serum. Here I've taken PBS, some water basically, and washed the inside of a mouse lung and collected the normal microbiota that grows in there and used that. So that's full of bacteria. And you can see that that alone is sufficient to make Titan cells. And if we take E. coli, we can do that. And E. coli don't even have to be alive to do that. And if we take another organism called Strep pneumoniae, we can do that. So all of this is saying that bacteria are participating in this behavioral change that Cryptococcus is undergoing. We were able to track it down to the individual molecule that the bacteria the fungus is sensing, and it turns out to be the cell wall component of bacteria, which is called peptidoglycan. And I'm showing you the structure of that here. It's two pieces, it's a sugar backbone, and then it's got a peptide side chain, an amino acid side chain. And if you just take half of that, the sugar and the peptide, that's moramyl dipeptide here, and that in itself is sufficient to induce these increased large cell sizes. So this is telling us that these cells that we've seen in these patients are actually the fungus responding to the bacteria that's in the host, the human lung, and undergoing this change to become these very, very large cells that are important for dissemination to the brain, redirecting the immune response, and causing disease. Okay, so hopefully you've now seen a little taste of this, which is that when we grow cells under the conditions that are more relevant to them, rather than our preconceived notion that cells should be happy and enriched media before they go into their stress conditions, we can have this huge amount of variety that can now tell us about the fungus. So we have, this is what we see when we put our cells with macrophages. So you've got these 5 different ways of growing all of a sudden. So here's an example of a macrophage in red that has engulfed Cryptococcus, and it's successfully killing the Cryptococcus. That's what we want to happen. In purple is an example of Cryptococcus that is being shielded by its capsule. And you can see, even though it's definitely in contact with that macrophage, the macrophage isn't responding to it. It's basically invisible. It's a cloak of invisibility. In green is Cryptococcus growing like it normally would in an extracellular space. So it's just budding and proliferating. And in yellow is a really unusual process called bombocytosis, which is where Cryptococcus actually starts out inside the macrophage, and then the macrophage kicks it out. So that's what you're seeing there, but the macrophage is still alive. You can see that. I'll just start here now. So it just popped out. And then lastly, we can actually see Titan cells forming in vivo and inside a macrophage. So that's what's in the blue there, and that light blue, you can see it's getting bigger and bigger and bigger, and that's the formation of a Titan cell in process. All right, so I told you I was going to tell you 2 stories, so let's get started with the second one. And that's going to be a story about an organism that has only recently been really described, which is called Rhizopus microsporus, and that causes a disease called mucormycosis. I need to talk, this is work that I did in collaboration with a number of people, but it's really the brainchild of Kirsten Volz, who closed her lab back in 2017, and her PhD student, Herbert Itabangi, who's a brilliant scientist now working in Uganda. And in collaboration with another PhD student, Poppy Sefton-Clark, and my collaborator, Jason King, who's an expert on soil amoeba, which will become clear in a minute. So this all starts with work that was done by Kirsten and a mathematical modeler named Sarah Jabari. And what they wanted to understand was, why is it that mucormycosis causes such terrible disease? What is going on with these? And they noticed that among patients who are suffering from mucormycosis, it's a particular category of patients. It's patients who have diabetes or patients who are on steroids. And these are patients that have a particular delay in their immune systems. So they're basically not able to fully respond to infections. And so they asked, well, is it just the ability to recruit macrophages to the site of infection that is causing that sensitivity? So this is an experiment they did where they have zebrafish. This is a zebrafish model of infection. And they infected these fish with either the fungus by itself, or they treated the zebrafish with steroids, like a high dose of steroids, like a patient might experience. And what you can see is that in the early stages of infection, the 2 systems are not different. You can't tell a difference. The systems look the same. So basically about 20% of the fish die early on. But then over time, these steroid-treated fish are much more sensitive to the infection. And what they noticed was that if they took the fish at 24 hours and they just counted the number of macrophages that were at the site of infection, they were able to predict who would survive and who would die. So you can see that here. If there was a very few macrophages, they would die quickly. And if there were many macrophages, then they would die later. So this is a hint that immune cells are really needed right at the beginning of these infections, or really, really early on. And I want to relate that to the what we see in global events in terms of mucormycosis, because if you know mucormycosis, it's because you remember it from a major outbreak that happened during the Delta wave last year, when in India, when we had the black fungus killing patients in India, right? And this was an awful, awful situation where in the course of about 3 months, we had 40,000 people come down with this disease very, very quickly. what it was traced back to was an underlying COVID infection that was exacerbating diabetes. And again, diabetes and COVID are both associated with a reduced ability of these patients to recruit macrophages to the site of infection, so they can't block the disease. So this is telling us that not only is this happening in zebrafish, but it's also happening in humans. So in order to understand what might be going on here, Herbert tried to model these infections. Rhizopus is a really hard system to study. It's not, there's no genetics that we can do in this system up until very recently. And a lot of what we know is based on analogy to other systems that we have a little bit more understanding of. So the one that we have the best understanding of is an organism called Aspergillus, which I think probably a lot of people in the audience are familiar with. And one of the things that's really interesting about Aspergillus is that it hides itself from immune cells with a layer on the outside of its cell, which is called the rodlet layer. You can see that here in dark blue. But when the cells are growing, when they germinate, they shed that layer and they become easily recognized by the immune system, which is why the most of us are able to deal with the environment, the Aspergillus in the environment. So if you look at the ability of macrophages to clear Aspergillus, what you can see is that in resting spores where they have this rodlet layer, macrophages aren't very good at taking them up, but as soon as they start swelling and they reveal themselves, now macrophages are actually pretty good at grabbing them and clearing them away. And if you remove that layer, so in what's called a PKSP mutant, which doesn't have that rodlet layer on the outside, now macrophages are really good at killing even just spores before they germinate. So the analogy that Herbert said, well, okay, we know macrophages are really important for the early stages of infection in Microsporus. We would expect that we would see the exact same thing in Rhizopus microsporus infections. So again, this is the prediction that we would make, that in Rhizopus, which doesn't have this masking ability, we would expect macrophages to be able to easily engulf them. But actually what Herbert saw was the exact opposite. As the spores swelled, he saw less phagocytosis by macrophages, less uptake. And in fact, the macrophages just looked kind of sleepy. They looked kind of sad. So what this is telling us is that Aspergillus is actually not a good model for this Rhizopus, because uptake of Rhizopus does not mirror these findings in Aspergillus. Swollen spores are taken up less readily. And so we wanted to know why, what's going on here. Herbert did a ton of work to try and figure this out, but what he actually tracked it down to was something that this fungus is secreting into the media. So if we take supernatant from this is just the liquid that the fungus is growing in, and we filter it, and we just treat macrophages with that media, and then we add particles, and we ask how good are these macrophages at taking up these particles, we can see that media from Rhizopus blocks the ability to do that. So you can see that here, where it's blocking the uptake of resting spores or even other fungi. So here's the ability to take up Candida albicans or Saccharomyces cerevisiae, which is just your normal bread yeast. So when we use this supernatant to treat the macrophages, now suddenly they are really not good at taking up particles in their environment. So there's something these cells are making that's blocking that ability. So what might be the source of this inhibitory molecule? And I hope if we were in a classroom that everybody would be raising their hands and saying, have you thought about bacteria? Because I basically primed you for that. And actually, when we start looking into that, what we can see is that bacteria produce really useful chemicals in combination with fungi. And there's some really cool examples of this. These are not just bacteria that are in the soil the way I've represented them, but they're bacteria that actually live inside these fungi and produce these molecules that help the fungi deal with their environments. So here's an example of the same fungus that I've been talking about, Rhizopus microsporus. And you can see this green is this bacteria that's living inside of it. And it produces a compound called rhizoxin. Rhizoxin is really interesting because it basically blocks the ability of cells to proliferate, to grow, because it blocks the actin cytoskeleton. Here's another example, a really beautiful example of another fungus called Mortarella, and it's the endosymbiont Candidatus mycovitis, and it actually produces a molecule that can kill other organisms that are around the fungus. So there's lots of examples of these molecules. So what did Herbert find? Herbert, when he looked more closely at this Rhizopus microsporus isolate that we had, he saw that it was chock full of bacteria. I mean, this is just like so crowded. And when we looked a little further, we were able to, number one, isolate that bacteria. So you can see that bacteria growing freely by itself without the support of the fungus. And also we can grow the fungus by itself without the bacteria. So this is a friendship, but not a partnership, but not an obligate relationship, right? This is a very transient and dynamic process. Okay, so why might this partnership, this transient partnership, be happening? So I mentioned before that this is work done with our collaborator, Jason King, who works on Dictyostelium. Dictyostelium are these little tiny amoeba that live in the soil, and they basically go around feeding on bacteria and fungi. And Rhizopus is an organism that doesn't normally grow in humans, it normally grows in the soil. And so we asked, maybe this partnership is actually helping the fungus survive in its interaction with these soil amoeba. So just to remind you, here's our example of our supernatant from our Rhizopus pair, our partnership, and you can see it blocks Candida uptake, it blocks Saccharomyces uptake, and it also is able to block feeding by these these little amoeba. So what we think is going on here is that this bacterial fungal partnership is actually protecting against predators in the soil. All right, so let's relate this back to the, why is this important? Well, so the fungus, what we were able to show was that when the bacteria is present, it helps the fungus go through its whole life cycle. So here's the ability of, so what we're doing here is taking the fungus and passaging it on plates over time. And you can see over time, when it has the bacteria in it, it makes a certain number of spores and it's very happy. But if it loses that bacteria, it starts making fewer and fewer spores. It's not going through its life cycle as effectively. It's also got a very different cell wall, and you can see that here. This is again transmission electron microscopy, like I showed you before, where you can see in the presence of the bacteria, it's got a nice thick black outer cell wall here, and this is much thinner in the absence of the bacteria. And that's quantified here over time. You can see that without the endosymbiont, different components of the cell wall have changed relative to with the endosymbiont. That's important because that cell wall is really involved in the ability of the fungus to resist stress. And you can see that here, where when we have reduced, when we lose the endosymbiont, now phagocytosis, so killing by macrophages, uptake by macrophages really increases, and killing by macrophages really increases. So this is survival of the fungus. So when it's in a partnership, the vast majority of the spores survive. When it's lost that partnership, the vast majority of the fungi die. And we can put that bacteria back in, and we're able to restore that partnership again. And that's, again, important for disease outcomes. You can see that here in zebrafish. So what we're looking at here is resting spores and swollen spores that have the bacteria or don't have the bacteria. So when we say plus endo, that's with the bacteria, and minus endo is without the bacteria. So when you're a resting spore, which is the dotted lines, you're basically, there's a delay in the ability of the organism to cause disease, but if the endosymbiont's there, it still kills the fish. If you're already swollen, if that bacterial relationship is already activated, now the fish die much more abruptly. And if you don't have that bacteria at all, and you don't have that head start, now the fish basically survive. So what about in zebrafish is very nice, but what about in mice? This is a little more challenging because we can't, we wanted to model these infections in patients, right? Because the goal here would be to be able to treat patients with an antibacterial at the same time as they're being treated with an antifungal to sort of help reduce the severity of the infection, right? So to do this experiment, we just co-incubated our fungi in the presence of antibacterial or not. And what I hope you can see here is that when they are treated with an antibacterial, after 2 days, the fungus is gone. But if they're not treated with an antibacterial, then they're more likely to be sick, to be infected. So overall, the message there is that removing the bacteria reduces fungal disease in both zebrafish and in mice. All right, so just to bring this to a close, I hope I've shown you that our viral particles, which can affect our immune status by either by making us HIV positive and then reducing our ability to combat cryptococcosis, or by reducing our ability to mount an effective innate immune response. So with macrophages being recruited at site infection can really increase our susceptibility to these fungal infections. And that patient samples and patient experiences with that have really informed and driven a lot of the basic biology and our understanding of these organisms. I just want to highlight that there's continuing need for research into fungal pathogens. We're looking here at percent mortality. So if you get an infection, although those infections are very rare, what's your likelihood of dying? So here's HIV. So, and again, lots and lots of people have HIV, but most of them are now surviving that infection. But if you have cryptococcus, your percentage of, your likelihood of dying is about 70%. So it's quite severe. And if you have mucormycosis, here it's approaching 90%. It's really a devastating infection. And one of the sort of worrying things of that is that even though we're really good at now treating HIV, progress treating other fungal infections has really plateaued. So this is the number of people who have died of HIV-associated infections in the last 10 years. And despite this 5.5 billion pounds being spent each year on treatment, we're still really fighting this problem. So there's continuing need for research. I'm going to stop there and thank all the people who were involved in this work and just open up the floor to any questions that there might be. Thank you so much.
Right, I'd like to thank Liz Ballou for that. That was an extremely interesting presentation, and I'm glad it's going to be recorded because I'm going to have to look at it again, I think.
I read out some questions. So if you have any questions, please do type them in the chat, but I'll go back to the first questions that were submitted. So this was when you were talking about the Cryptococcus, Liz. So first of all, from Jorge, is there a kind of intelligence in a fungus? And this was in relation to the ability of the fungus to sense its neighbors.
You know, that's a big argument in the community, I think, because what is intelligence, right? Is the ability to predict what's going to happen next? Is it the ability to understand your environment? These guys don't have brains, but they do have very complex signaling systems, and these signaling systems have evolved over, you know, millennia to respond to very predictable signals. And I think that's what's really interesting to me about the bacterial fungal interactions, because before there were people, before there were mammals, there were bacteria, right? And so the relationship between fungi and bacteria is ancient, and we really don't know very much about it, but it's clearly affecting the biology of these organisms.
Okay, thank you. Again, a question about when you were talking about the Cryptococcus from Philippe. If you delete the quorum sensing pathway, would they become Titan cells regardless of cell density?
Okay, so this is something that's driving me nuts because the answer is no, right? You would think that if they can't tell that they're in a crowded environment, that they would all be big, but there's something more there. There's something else going on. So we're working on that problem right now to try and answer it.
Okay, so a question from Jan. Jan says, amazing work, Liz. Do you think that the few neighbors concept also contributes to titanization in vivo? And do you see titanides in vivo?
Yeah, okay, so yes, I do think that lower density infections in vivo are related to more cells. That's a really difficult question to ask because we're even at very high levels of infection, we're still quite low density in the lung environment, right? But what we do see is if we infect mice with large cells, then they tend to have more brain infections. There's definitely something going on there that's related to what's happening in vivo. We also do see titanide, these little tiny cells in vivo as well. So I have lung sections where we're able to see, yes, we have these oval cells, they have a very thin cell wall. And you may or may not be familiar with another cell type that people see in vivo, which is called the microcell. These are, again, are really small cells. They're about 1 micron, but they tend to be round and they have a very thick cell wall. So they're quite distinct from these titanides.
Okay, thank you. And then moving on to your second story about the rhizopus. So again, from Jan, do you know the signals produced by the bacteria that promote cell wall integrity of the fungus and hence stress resistance?
That's what we're trying to figure out right now. So we just got a new postdoc in the lab, Dora, and the goal of that project is to try and understand what exactly is it about that relationship. There's sort of two hypotheses there. One is having something live inside you is stressful, right? And so because you're mildly stressed, you're always going to be mounting a little bit of a stress response, right? And the readout for stress response is a thicker cell wall. So it may just be that the sort of presence of any molecule inside of the fungus will trigger this response. The more interesting one is that there's actually a direct interaction between the bacteria and the fungus that is supporting that. And there is evidence that bacterial gene products can influence fungal transcription. So there's transcription factors that come from the bacteria that go into the fungal nucleus and can change the behaviors. So it may be a very simple explanation, and or may be a more entangled one.
Okay. A question from Linda. Can we expect to see these same interactions in plant diseases in relation to rhizopus as a root disease and Cryptococcus in oak trees?
I don't know about Cryptococcus in oak trees.
That was Cryptococcus only?
Yeah, I don't know. That's a good question. I don't know about that, but I do for sure rhizopus and endosymbiosis influencing plants. So, for example, the bacteria that we found is called Ralstonia piquetti, but it's a close relative of an organism called Ralstonia solanacearum, which is a pathogen of vegetables and tomatoes in particular, so plant wilt. And there's clear evidence that there's a bacterial fungal interaction there that supports the pathogenesis of that system. The mechanism may be different, but yeah, for sure these relationships are also influencing plant immunity. I think you're muted, sorry.
Sorry.
Okay.
Thank you. There's another question from Linda. Does predisposition to titin cells caused by the presence of a viral disease in a human result in greater threat to a sufferer in the natural bacterial-rich environment?
So there's a lot there that you just asked. The first is that not all clinical strains of the fungus make titin cells. And that's one of the questions that we're working on in my lab and in other places. And the link between titin cells and disease is one that's still not clear. The question of whether or not having a viral infection interacts with that process. Again, we can't say for sure that, for example, patients who have HIV have more titin cells because there might be a connection with the particular bacteria, the fungal isolate that you're working with there. But I guess I would say that having co-infections is dangerous in general for patients, right?
Okay, thank you. A question from Paul. Do the titin cells have just a single nucleus? If so, and if the size increases mainly for defense, any ideas why they become polyploid? I.e., what is the polyploid advantage here?
That's a great question. So I didn't go into this at all because it's really complicated to explain to a general audience, especially when you can't have a real conversation like would be over in person. But basically what's happening is when cells convert from yeast to titin, they go through their cell cycle, their nuclear cell cycle multiple times without going through their budding cell cycle. So they get bigger without producing daughter cells. And while they're doing that, they're making more copies of their nuclear content. It's a single nucleus. So yes, it's called endo-reduplication rather than cell cycle duplication. And the advantages of that are that, well, first of all, you have all your nuclear material ready to go. So when you do make the choice to start making daughter cells, you can go much faster, right? And we see that their cell cycle time is cut in half because they don't have to duplicate their genome. But when we look at the daughter cells of those mother cells, we see some unusual features. So one is that they tend to be aneuploid, meaning they have extra copies of their genome. Or they're diploid, meaning they have 2 copies of their genome. And the hypothesis is that genetic diversity, that genomic diversity, allows particular members of that population to be more resistant or ready to deal with new stresses that might be encountered. So it's a way of generating population diversity really quickly in the face of particular challenges. And you might think of nutrient starvation as a challenge, or bacterial invaders as a challenge. One of the things that we see that's really interesting is that these daughter cells tend to be associated with drug resistance. And as you know, many antifungals are actually bacterial products, right? So you can think of an example there where this is a way to quickly evade and develop new resistance to compounds that are in the soil.
Thank you. That's all the questions that have come through on the chat for now. So if anybody has got any more questions, there's a minute or two just to quickly type them. But just thank you again, Liz, for a fascinating talk. That is one that I think, as Norman says, I'll need to watch again. So we have been recording. So the good news is it will be up on the BMS YouTube channel shortly. So keep an eye out for that. Anybody who wants to watch again.
Great. It's really been my pleasure. Thank you so much for giving me a chance to share my work with you guys. It's a great opportunity.
Good.